Hi, APE students. Welcome back to the third College Board virtual session where we're going to hit up a couple of more of the topics in Unit 8. Just remember that although Unit 8 isn't formally assessed on the exam, we want to make sure that you get a little bit of exposure to the content so that you feel that you've gotten the full depth and breadth of your AP Environmental Science course. There's again some pretty great topics yet to cover that you might see in your college AP Environmental Science studies, or if you go on to take another AP Science exam. So my name is Amy Fassler. I teach in Northern Wisconsin at Marshfield High School. This is our first day of spring break today. We're kind of late, so we're going to start our warm up with a little virtual spring break time, going back to the nice warm salt marshes and coastal ecosystems and mangrove forest that Dr. Sowell talked about yesterday. So let's get going and take a little bit of time to review that, and then we'll get into endocrine disruptors and some of the topics for today. So Dr. Sowell yesterday gave us some beautiful examples of the many valuable ecosystem services. And remember, that concept is very global. I could be asked in an FRQ to identify an ecosystem service of just about any ecosystem. So if I were asked to do that, to identify an ecosystem service, one of the most important examples Dr. Sowell gave yesterday that jumped out at me was their ability to absorb and filter runoff, and therefore improve aquatic water quality. The next part of a prompt might ask me to explain why that is an important service. And remember, when the skill or task verb asks me to explain why, I'm providing a reason for something. So sometimes those two prompts might even kind of be blended together. And I often like to instruct my students to put those transition terms in there. So this is an important function because it could prevent nitrates from entering the coastal ecosystem. And I want to keep going a little bit by adding just a little bit more. And again, if I have that transition word, therefore, I reduce the chance of algal blooms in hypoxic zones. You sometimes know that complete thought to make sure that you've really explained why that's an important function. Because if I just stopped at they prevent nitrates from entering the coastal ecosystem, my question to you would be, well, what's my problem with nitrates in the coastal ecosystem? So that because and therefore are really nice prompts to keep you to get that entire explanation of why that's important in this case. Sometimes on the AP exam when we're reading, a really common type of student response to this might be something like, well, the pollution goes into the water and that's really, really bad. That student understands that in the idea that that process, that there's something happening within that ecosystem that's absorbing those nutrients, but you got to put a little bit of specific information with that. When you're asked to provide or explain why a particular pollutant that enters the ecosystem, what kind of impact it's going to have, you can't ever just say a pollutant. We need a little bit more specific there that demonstrates your knowledge of what you've learned in the course. And the same thing with explaining why. My explanation can't just say because this is very harmful. I need to make sure that my explanation is specific to, in this case, the pollutant I added, which was nitrates. All right, so we're kind of back into the idea of aquatic pollution. We're going to go ahead and take an overview, look at an overview of what we're going to do today. We're actually going to cover a little bit more than endocrine disruptors. We want to make sure we get through kind of the big ideas in the units 8 and 9 in the limited time we have. So I'm going to combine 3 different learning targets: persistent organic pollutants, endocrine disruptors, and bioaccumulation and biomagnification. And one of those topics really lends itself to the discussion of the next one. They flow very nicely together. Just like in the first couple lectures we've had, we related a lot of content back to Unit 5 on land and water use, because aquatic pollution a lot of times comes back to, what am I doing? What is the impact of urbanization, or clear cutting, large scale logging, or habitat fragmentation, or mining, or industrial agriculture, or fish farming. So I want to be able to take those ideas, and we're going to expand on that a little bit. There are a number of different skills that we'll focus on. A couple of those look familiar to you. Some of those you saw with Dr. Sowell. So let's go ahead and get started by looking at persistent organic pollutants. I'm going to introduce some new content here. It might be new for some of you. And there's a little bit of background information that we need, some vocabulary we have to kind of break down. So just sit back and kind of chill for a little bit while we walk through and start this story of these toxic chemicals and what happens when they enter the environment. So when I look through my couple of essential knowledge statements here, this is really describing for me what a persistent organic pollutant is. This is talking about kind of relating that description and some of the chemical characteristics of that substance into it's being fat soluble, and that property of those persistent organic pollutants is how we explain biomagnification. So let's break down a couple of the terms that we've got here. The first term is that this big class of compounds, this big class of toxins that can enter the environment are persistent. That means that they're not going to break down or degrade readily once they enter the ecosystem, or once they enter the body of an organism within that ecosystem. It's kind of like your little brother or sister who's always poking at you, asking you for some help or come play with me. They're very persistent. They won't go away. So these are compounds that once they're there, they're going to stick around a long time. Next, these are compounds that are organic. That means that they contain carbon. I just happen to have a molecule of glucose here. You guys looked at organic compounds in the unit on ecology when you did photosynthesis. So here's an organic compound. This is a molecule of glucose. Started out as inorganic CO2 in the atmosphere. Through photosynthesis, plants made this amazing molecule of glucose. These are each an atom of carbon. I've got 6 oxygens and I've got 12 hydrogens. So this is an organic compound, but it's not a persistent organic chemical. We have obviously a lot of natural organic compounds, any kind of sugar, glucose, starch, any carbohydrate, cellulose, the material that plants use to make their stems and the structural components of their plant bodies, proteins, lipids, nucleic acids. So we're a carbon-based life form. There's lots of naturally occurring organic compounds. But when we talk about persistent organic compounds, we often talk about compounds that are, we're referring to compounds that are synthetic. So these are where I take carbon atoms, that's my raw material, to build this synthetic compound in a laboratory. All of those carbons come from petroleum refining in the petrochemical industry, and organic chemists take and tweak those molecules. So remember, I'm talking about things like everything from methane that we burn in our natural gas furnace, to octane, 8 carbons long, that I put mostly in the gasoline in my gas tank. And so chemists take those different fractions of crude oil and they manufacture synthetic organic compounds. Plastics are a synthetic organic compound. If you look at the labels of your clothing and you see any fiber like polyester or rayon, those are synthetic polymer compounds that ultimately came from crude oil. A lot of times when we talk about the synthetic organic compounds, they are present in their ring structure. It's a little hard to see here, but this is that straight chain glucose molecule that we had. And a lot of times we see that represented in sort of this ring structure like this, where, I mean, for glucose, there's an oxygen in my ring here, but otherwise all the members or parts of that ring are carbon atoms. And we'll look at those again in just a minute. What else describes a persistent organic pollutant? They're mostly non-polar fat-soluble substances. And this has a lot to do with that part of the essential knowledge statement was how do they get transported in the environment? Once they enter the environment, how do they get in my body or the body of an ecosystem? And what is the impact of that? So that's where we really look at the idea of biomagnification and bioaccumulation. And then they're particularly harmful because they have such a great ability to be transported in the environment and many different ways that we can get that exposure. So let's dig in a little bit deeper to these persistent organic compounds. And to do that, we got to review just a little bit of chemistry. So remember my description in my knowledge statement said that they're mostly non-polar fat-soluble molecules. Well, let's contrast that with water that's polar for a minute. If you remember the basic geometry of the water molecule, I'm going to get to the big idea here, so just bear with me. What we really care about when we say this water molecule is polar is that it has poles or ends to it, like the north and south pole of a magnet or the north and south pole of the earth. There are different ends to it. This is the oxygen. Remember, we saw the little red oxygens in our glucose molecule. The oxygen end of the water is a little bit negative. It has a pole or a negative end, and the hydrogen ends are a little positive. Why do we care? Like, what does this have to do with these persistent organic chemicals? Well, if I have a chemical that I put in water, anything, we call water the universal solvent because it has those charged ends to it. Remember, opposite charges attract. So whenever I put something into water that has a charge to it as well, it's going to stick to that water molecule. It's going to go into solution. Non-polar substances, this is a big molecule of a fat. This is like a triglyceride. This is the fat like I would eat if I ate a big cheeseburger and a lot of fat in it. So this is what that molecule looks like. And although we've got some kind of different stuff going on this end here, this is what I really want you to focus on. All of this molecule is non-polar. This is just a bunch of carbon atoms with hydrogens all around it. All those electrons are evenly shared. There's nothing going on there where I have an end of this molecule that is positive or negative. We say it has no poles, it is non-polar. And this is a molecule of hexane. And just to show you, that's 6 carbons. It's kind of like glucose, except it doesn't have the oxygen, but it's 6 carbons in a straight line. And instead of these OH groups coming off of here, those would all just be hydrogens. And that is a product of petroleum as well. So let's see if we can visualize what I mean when I'm saying that something is soluble in fat, and then we'll make sense of why that's important. So let's see, here's a little bit of water. You probably know that oil and water don't mix. Because they have different polarities. So when I put a little bit of oil into the water, that oil is going to form a layer on top of the water. Can you see that? There it is. See that oil is forming a little bit of a layer on top of the water. That oil did not go into solution in that water. Well, what if I put a little bit of oil in there with a little bit of hexane? This is hexane. This is that molecule that you see here that is also nonpolar. And so when I put the oil and the hexane together in a test tube, they do not form a layer. Remember, my oil and my water form a distinct separate layer. They are not soluble in each other because there's nothing on this molecule to make it stick to the water molecules. But when I have 2 things that are nonpolar, then one can go into solution. And so if you think about how this relates to toxicity, if I get exposed to a toxin in the environment through the food that I eat or my water supply, or I breathe it in through the air and it's water soluble, It's going to dissolve, and I've got a lot of water in my body. And even though that substance goes into solution in the water, like Kool-Aid goes into solution, and I stir it up and I can't even see the Kool-Aid anymore. So that water soluble toxin gets dissolved and it's carried around through the water in my body. But it might impact my body as it's being processed by my body, and maybe the liver's detoxifying it a little bit. But ultimately, I pee that out, or I sweat that out, or I excrete that because it's soluble in water, and I excrete water every day through my digestive process. So it might have a little bit of an impact on me, but it's not going to be stored in my body. My body's going to flush it out when I pee. If I have a toxin that is fat soluble and it dissolves in that fat, I don't pee fat out every day. Think of all of the parts on your body where you've got fat stored. So if that fat soluble toxin, like the hexane, went into solution in the oil, that fat soluble toxin is now with my fat. It's being stored, and my body is not going to excrete it or expel it. So let's just kind of review what we've covered so far. We have these chemicals that are synthetic organic chemicals. They're made with carbon atoms from petroleum in a chemical laboratory. They are persistent, they don't break down, and they're fat soluble, so that my body is going to store them. It's going to accumulate in fatty tissue versus being expelled or peed out in my urine. So let's just take a quick check on that. This is a multiple choice question, but I really like it because notice there's still a command term here. It's still asking me to describe a way that persistent organic chemicals can be harmful. That's exactly how a prompt might appear on an FRQ. So it's still good practice to look at those and pull out a good description. I'll let you have just a second to look at that. Oh, that should have been a pretty easy one. You probably wouldn't have known that 10 minutes ago before we started our discussion. So letter C, persistent organic pollutants are soluble in fat, so they accumulate in fatty tissue. Hold that thought because we're going to come back to it in a little bit. So let's look at a specific example of a persistent organic chemical now that we've kind of described them, and hopefully have a pretty good feel for what they are, and kind of something about their chemical nature that's going to help us be able to understand the impact that they have. And you've probably heard about DDT back in Unit 5 when you talked about pest control methods, and you looked at the idea of pesticide resistance and the pesticide treadmill. So here's the molecular structure of DDT. They're never going to ask you to know that, but you now, even with our limited discussion, should say, aha, I see that there are carbons in a ring structure here. That's what this little kind of shorthand symbol represents. Each of these indicates a carbon in that ring. They're held together by double bonds. All we really care about is that I can see that this is an organic compound. Right away, if I'm talking about toxicity, my brain is going, there's a good chance that that's a persistent organic compound. So in our discussion of pesticides, you may have been introduced to Rachel Carson, and I'm going to play just a little bit of a clip because you're going to see how chemists first started making some of these organic compounds in the lab back after World War II.
Pesticides weren't always thought to be harmful. To the contrary. In 1947, Time magazine carried an advertisement claiming DDT was good for people, homes, and farms. It took 20 years before scientists realized how dangerous it was.
It begins with the war-born development of DDT. This diabolical weapon of modern science saved millions of humans, but killed billions of insects. Man, with this newly discovered force, has at long last gained the upper hand in our age-old struggle. Just like these mosquito larvae, it came from laboratories where top scientists from famous universities and from industrial and government organizations collaborated to develop something new and different. They succeeded. They perfected Pestroy, the most effective weapon man has ever wielded against insects.
So one of the benefits of DDT is that it was really useful in controlling pests, and it reduced crop loss or increased crop yield, so that farmers could make a profit and so that we could grow food to feed a rapidly growing human population in the United States baby boom, for example, after World War II. Rachel Carson first raised the alarm about the potential impacts of DDT in her book Silent Spring. And she sort of talked about that idea of the pesticide treadmill. Remember, our goal in using pesticides is not to kill every insect. It is to reduce the amount of crop that I lose. I expect a little crop loss, but not so much that I still can't make a profit. So Rachel Carson believed that there may have been a role for pesticides in controlling insect pests that reduce crop yield. But like any good scientist, she started asking questions, just like we want you to ask questions, both as young scientists and consumers that may be exposed to these persistent organic chemicals. Is there a certain dose or concentration that's safe? What happens to that DDT after I spray it out of the airplane and it lands on an aquatic ecosystem? So Rachel had a lot of questions, many of which were answered or addressed as a result of the publication of her book. So let's take a second and see if you can identify something about DDT as a persistent organic chemical. Again, this is looking at the task verb describe. So if on an FRQ, you were asked to describe something, you are listing characteristics of it. Take a second and look at that one. And the correct answer is A. Notice that statement B is a correct answer, and they alluded to that a little bit in that film clip that we just watched. They talked about the benefits of it, and there are benefits both from reducing crop loss to killing vector insects that transmit really harmful diseases that could kill large numbers of people. Things like malaria, typhus, Zika virus, and we'll be looking at those vector diseases a little bit more next week. Obviously, the correct answer is that it's very stable. And so DDT is going to stay in the soil or whatever ecosystem it ends up in for a long period of time. Okay, so hopefully you've got some pretty good general idea of what those persistent organic chemicals are. Stay around a long time, have carbon, and they're fat soluble because they're nonpolar. They've got those carbon in rings usually because that ring structure is more stable than the straight chain. Okay, so now we can get into endocrine disruptors. This is one of my favorite topics. Endocrine disruptors, and we often call them estrogen-like chemicals. I'll show you that term in a minute. They are a type of persistent organic pollutant. So I already know a lot about it just by knowing it's a persistent organic pollutant. I know it's going to stick around a long time. I know that it's a synthetic compound that's manufactured in a lab. And there's a likelihood that it's fat soluble because most of those organic compounds are kind of fat soluble. So let's look at our knowledge statement. The first knowledge statement describes an endocrine disruptor. They interfere with normal endocrine system. Okay, we'll explain that in a minute. And then this is the impact that they have, and we'll look at that as well. And we'll look at, again, a couple of those skills or practices. So here is kind of a simple diagram that explains how endocrine disruptors work. First of all, the endocrine system is our system of glands that produce hormones. And when I look at what they regulate, I could really have made that list even 10 times longer. We have so many different hormones that do so many different things. They regulate metabolism. That's like the way my body processes and absorbs different nutrients, growth, development, sexual reproduction, development in the embryonic stage is influenced. We now know that endocrine disruptors can influence everything from aggressive behavior or learning disabilities. So the list of things that are controlled by our endocrine system, the number of hormones that control different processes is just really a big list of things. So endocrine disruptors have the potential to disrupt a lot of different activities. What you kind of want to think about when we think about hormones, like insulin is a hormone you've probably heard of. And what insulin does is there's a signal that released if things are working right in my body, that if I just eat a diet that's got, or if I just ate a lunch that was really high in simple sugars and the amount of glucose floating around in my blood is really high, insulin says, oh, wait a minute, I'm going to go gather up some of that glucose and I'm going to pack it away in a compressed glycogen form, and I'm going to store that away in case this person doesn't get any food for a while, and then I can release it and they'll have glucose for energy. So you can think of these hormones as like little switches that turn a process on or turn a process off when everything's functioning the way it should. So here, for example, is my cell. And when a normal molecule comes along, like an estrogen like chemical, and I'll show you the real molecule of that in a minute, there's a certain end. You may have looked at, like, have been seeing images of the coronavirus, and they talk about those glycoproteins, those protein spikes that are coming out of it. Well, it's pretty much the same idea of what happens when a hormone has to bind on the surface of my cell. Here's my normal estrogen hormone. Here's my cell. There's a perfect fit there. And the minute that estrogen binds in that cell receptor site on the surface of the cell, it's kind of like it opens a door. It turns a key, and a whole process of other chemical downstream reactions can start to happen. And you may recognize the hormone estrogen. It's the hormone that stimulates the development of secondary female sex characteristics. And it's important for determining cycles of ovulation and release of eggs and implantation of eggs. It's a very powerful hormone that controls a lot of reproductive functions in my body. So let's say, for example, and I'm going to show you these images now, you may have heard of the chemical BPA. The chemical name for it is bisphenol A. That's why we call it BPA. And you may have a water bottle that literally is labeled, oh, just so you know, this is BPA free. So BPA is a persistent organic chemical. It is one example of an endocrine disruptor. Persistent organic chemicals, endocrine disruptors are one sort of big group of organic chemicals. There are other things like DDT as well. So here is that molecule of estrogen that just bound on the surface of my cell. Here's the molecular structure of it. Here's its little skeletal structure. We recognize now that this is a ring of carbon atoms. So simply knowing that, I know a lot about this. I know it's probably persistent. I know it's probably fat soluble. I know it's something that doesn't occur naturally anywhere on, estrogen is a naturally occurring hormone that works in developing secondary female sex characteristics. Here's the molecular structure of BPA. And again, now I already know a lot about that. I know that it's carbon. I know it's organic. I know that if it's a persistent organic compound, this doesn't occur if it's a synthetic compound and it is man-made. This doesn't occur anywhere naturally on our planet. And that inherently should kind of be raising some red flags to you, because you know how fine-tuned ecosystems are and how they've evolved to work well with what they're given. So this is this synthetic compound. And when I look at it, I'm going to give you just a second to look at the structure of BPA, the synthetic compound that I use in plastic. It's added to change the properties of that plastic, make it, I think, more durable. And here's the structure of that natural hormone, very powerful hormone in reproduction, estrogen. So when I look at them, it appears as if they've got some little OH groups, hydroxyl groups coming off them here in kind of the same place. So if I look back at that original model we were looking at, here's the way estrogen-like chemicals work. Here's my estrogen that bound in there really tightly. Maybe it was this OH group that bound in that cell surface and it made a really tight fit, and that there was a chemical reaction that occurred when that hormone binds on that cell surface. Now, here's my BPA, and it's got some pieces off of it that look kind of like estrogen. So for all estrogen-like chemicals, kind of two things can happen. Either this BPA molecule can kind of try to fit on that cell surface, and it doesn't quite fit good enough, but it's blocking it so that the real estrogen can't get in there. Or perhaps it's got enough similarity in chemical structure to an actual hormone that it can actually bind in there. And my cell thinks that estrogen is binding, and my cell is turning on all the processes that would be turned on just like if estrogen had bound there. Very powerful secondary development of secondary female sex characteristics. So let's see if we can bring this full cycle to the impact then on ecosystems. So here's a graph. Wanted to make sure we got a little graphic interpretation in. One of the biggest impacts of DDT, for example, on ecosystems was the thinning of eggshells. Remember that DDT molecule looks a lot like an estrogen molecule, and DDT had a big impact on, in this case, an osprey. Oftentimes, birds that are at the top of the food chain, tertiary consumers, fish eating or meat eating birds, for example. And if you think about it, the process of producing that eggshell to care for the fertilized egg, all is going to be, all of the processes that are involved there are stimulated or initiated through estrogen binding on a cell surface. Now, I'm making this sound pretty easy, but you're getting the big idea. So what happened when we used a lot of DDT, and that DDT ended up in an aquatic ecosystem, and a fish ate that, and then the osprey ate the fish, we saw that there was a great reduction in large bird of prey populations because of eggshell thinning. And this graph shows DDE. DDE is what we call a metabolite of DDT. Remember that even if that substance is somewhat water soluble, it's going to still have impact on my body as it's going through it. DDE is the sort of the breakdown product of DDT. So if I'm going to do an experiment to assess the concentration of DDT exposure, it's very easy for me to actually measure the concentration of what's excreted, if you will, or the product that broke down. It's sort of an indirect way of assessing DDT exposure. And the point that I really want to make here is this negative relationship, because this is sort of a misconception that a lot of students have about a positive and negative relationship when they look at information represented graphically. So this is my independent variable. The amount of, we're just going to say DDT, the amount of DDT determined the thickness of the eggshell. And I can see that as I increase the concentration of toxin, I decrease the thickness of the eggshell. I've got a nice trend line on there showing me that there is a negative relationship. There's still a relationship, but increasing the toxin doesn't increase the thickness. Increasing the toxin decreases the thickness. And as you can imagine, if the eggshells were very thin, the developing chicks just never reached it to maturity, and really harmed a lot of populations of large birds of prey. One of the other really interesting, unfortunately interesting, but kind of scary thing that we're also seeing with endocrine disruptors is feminization in amphibian or fish populations. Any aquatic organism, and we're going to come back to why aquatic organisms in a minute, But amphibians are really good indicator species because they live part of their life cycle on land and part in the water. They are very sensitive to pollutants in their environment and changes in the concentration of those. So what happens when these strong estrogen-like chemicals enter the aquatic ecosystem is that those amphibians or fish are getting very large doses and they are losing their masculinity. We've actually seen examples of there's almost no males left in this species anymore. And this is kind of what Rachel Carson started doing with good science work and what we as good scientists want to do. We don't know all of the impacts of these endocrine disruptors, but we certainly have good questions we want to ask. If a vertebrate animal, if an amphibian is being impacted at a certain concentration of these endocrine disruptors, can they function the same way in a human? That's certainly a good question to ask. Much of that exposure comes from pharmaceuticals, and Dr. Sowell is going to talk about waste treatment tomorrow. But even if I don't throw my pharmaceuticals, like my leftover pills, I could throw them into the river or I could flush them down the toilet. There's still another way that they could enter the ecosystem. Anybody think about that? Even if we're not directly or indirectly dumping leftover pills, they could be coming out through my urine, or the metabolites, the breakdown products of those things could be coming out in my urine. So this is a difficult problem to address, and one of the things that you'll look at in sewage treatment. Let's just take a quick how do you know what you know here and look at a question on endocrine disruptors. Which of the following best describes the action of an endocrine disruptor? And you should have been able to pick out letter B. They block the receptor protein binding site. That part we didn't go into a lot, maybe a little bit, so that the hormone can't receive a signal. So remember, that can either bind in there and act just like estrogen and turn that on, or it can block it and prevent in this case, estrogen, but it could be other hormones as well. These are far-reaching compounds. I just focused on the estrogen. So let's wrap up our new material by looking at bioaccumulation and biomagnification and bringing these concepts all together. Notice here that we've already talked about DDT. PCBs are kind of like BPA. They're another persistent organic compound. So the last thing we want to look at is mercury. And you've got the essential knowledge statements, which are basically a description or a definition of bioaccumulation, which is the buildup of a toxin in an individual person. I'm taking in a toxin at a rate greater than I can pee it out or expel it or excrete it. So it's accumulating in my body. Biomagnification implies that as that fat-soluble toxin as another organism eats that, or down-level trophic level organism in the food chain, because that toxin is fat soluble, each organism above it, its concentration is magnifying. It's kind of a hard thing to visualize. So I'm going to show you that in just a second. Let me just first show you where that mercury comes from in the environment. Burning coal, you looked at that in your energy and your air pollution unit, releases the mercury. The mercury often falls with precipitation or as dry deposition, falls on a lake, an aquatic ecosystem, and down in the sediment of that lake, there are microbes, there are bacteria that are converting that to a methyl mercury. Oh my God, they're putting that mercury on the end of a little organic molecule. Now it's fat soluble. So now when this little fish is eating some algae in there, it's getting that mercury now is an organic compound that's fat soluble, and it can accumulate in that fish. That big fish is eaten by, the little fish is eaten by a big fish, which is eaten by a human. And what happens in the process of biomagnification is that concentration increases as I go up a food chain. And this should look super familiar to you. Remember the 10% ecological efficiency rule, that these zooplankton only get 10% of the energy from the trophic level below them. Well, let's take that idea and relate it to the idea of biomagnification. So here are some little zooplankton, phytoplankton, fish, eels that eat the fish, and there's that osprey at the top of the food chain. I'm going to let you watch this quick little simulation where each M&M is going to represent 1 part per million of methylmercury. So the whole way this works is that each of those little zooplankton eat 5 phytoplankton. Each of the fish are eating 5 of the zooplankton, which each of the eel are eating 2 or 3 of the fish, and the osprey ends up eating both eel. Notice how pretty quickly the osprey got all of that persistent organic chemical. It got all of that mercury that worked its way all the way up through the food chain. It biomagnified. So as we're wrapping up, here are just a couple sources of some of these toxins. I did want to point out breast milk because that's something people maybe don't think about. But if I'm a human, a meat-eating human, a meat-eating nursing mother, I could be, if I'm eating fish that have a high concentration of mercury, my breast milk that I'm feeding my child may have a very high concentration of mercury. And breast milk is actually a really great human fluid, if you will, to use for studying toxicity, because I can get samples of it pretty non-invasively. And the nice thing about it is I can get toxicity exposure for both the mother and the child that's consuming that breast milk. So let's wrap up with just a little bit of guided practice. Identify one human activity that releases mercury and describe how that mercury is transported. And finally, explain why there are greater health risks associated with eating large predatory fish. These are both pretty much free response questions that we've seen before. Burning coal releases mercury into the environment. A mistake that students often make is they don't burn a fossil fuel. So don't just say using coal, because if I have a hunk of coal sitting here, it's not releasing any mercury. Mercury is transported. Remember, I'm describing a process, how that happens, and falls onto water or soil. And then the mercury can biomagnify or increase in concentration through the food chain. I think we're about out of time, and so I'm going to leave this practice math problem up here. You can go back and check it. The one thing I wanted to point out when you look at your dimensional analysis work here, please make sure that whenever you're setting up your dimensional analysis, that you have a unit that gets canceled. That's one of the biggest mistakes students make. And I think we hit a lot of those targets. Sorry, we went through that mathematical calculation pretty quickly, but I wanted to make sure and tell you thanks for coming. If you take an advanced environmental science class in college, or just being a good consumer, be a good scientist, ask good questions, and tune in tomorrow when Dr. Sowell is going to take you back to wastewater treatment plants and look at how we solve some of these problems with toxicity. Thanks, guys.